We have already discussed how the earliest life might have begun as chemoautotrophs and made glucose and ATP by chemosynthesizing inorganic chemicals in the early atmosphere, in the oceans, and discharged by volcanoes and volcanic deep ocean hydrothermal vents. What we have barely mentioned is how 99.9% of glucose is made, and that is by photosynthesis. We don't know accurately when the first life began. Life could have started anywhere between 3.8 and 4.3 billion years ago. What we do know is that the earliest confirmed fossils we have are of stromatolites that lived 3.48 billion years ago in what is now Australia. Stromatolites are clumps of rock formed by colonies of cyanobacteria. There may be other types of bacteria that developed photosynthesis earlier than cyanobacteria. The green sulfur bacteria family may be older than the cyanobacteria. As the earliest known photosynthesizing organisms, currently existing species of green sulfur bacteria include Chlorobium tepidum and many more. As their name suggests, green sulfur bacteria exist in sulfur springs and use sulfides including hydrogen sulfide H2S as its metabolic electron acceptor rather than oxygen O2. There are also types of purple sulfur bacteria whose ancestors may predate the existence of cyanobacteria. I focus on cyanobacteria as ancient photoautotrophs because cyanobacteria may have created the oldest known fossils, ancient stromatolites, and it is widely believed that cyanobacteria are the ancestors of the endosymbiotic chloroplast organelles that do all the photosynthesis in modern plant cells. Cyanobacteria were once called blue-green algae, but that term is defunct as the word algae is restricted to eukaryote algae. Cyanobacteria are prokaryote bacteria. Cyanobacteria lack the separate nucleus and mitochondria that define eukaryotic organisms. What these cyanobacteria do have is chlorophyll. Chlorophyll is the almost magical molecule that allows photosynthesis, the splitting of water molecules with light photons. Whether you call what chlorophyll does magic, or a miracle from God, or just plain physics is up to you. What the chlorophyll does is provide 99.9% of life on Earth with a food source directly from sunlight. Without chlorophyll, life could not exist on Earth, except for the handful of species that get their food from chemoautotrophs that live off chemicals expelled by volcanic deep ocean vents. Chlorophyll's splitting of water with sunlight is the start of the photosynthesizing process that makes the simple sugar glucose C6H12O6 from water H2O and carbon dioxide CO2. 6H2O plus 6 CO2 plus light energy reacts to form C6H12O6 plus 6O2. Although this overall photosynthesis equation looks simple, the photosynthesis process is a highly complex multi-step process. The photosynthesis process was present in the first cyanobacteria that lived 3.4 billion years ago. It is believed that cyanobacteria were first incorporated endosymbiotically into a eukaryote cell sometime between 1.8 billion and 600 million years ago. Since their incorporation, the engulfed cyanobacteria have evolved into the chloroplast organelles inside the cells of plants and algae, along with a few single-celled protozoans like the euglena. The chloroplast is where the whole photosynthesis process takes place. The structure of the chloroplast consists of several basic parts: the inner and outer membrane, thylakoid bodies, and various fluid spaces, the intermembrane space, the stroma, and the thylakoid lumen. A stack of thylakoid bodies is called the granum. Pay special attention to the stroma and the lumen. The stroma is the cytoplasm fluid of the chloroplast. The lumen is the fluid inside the thylakoid. The thylakoid bodies have their own membrane that separates the stroma fluid from the thylakoid lumen. Embedded within this thylakoid membrane is the electron transport chain that contains the chlorophyll that makes photosynthesis possible. The figure shows the labeled structure of a chloroplast. The chloroplasts may have some rudimentary DNA from their cyanobacteria ancestors. The thylakoid bodies have no DNA. Still, as the thylakoid bodies have their own membrane, electron transport chain with an ATP synthase motor, and cytoplasm lumen, we can question whether the thylakoids were once independent microbes that were engulfed symbiotically by the ancestors of cyanobacteria over 3.5 billion years ago. To enable cyanobacteria to enjoy the benefits of the photosynthesis system the thylakoids already had developed? No matter we can solve today. Since cyanobacteria were engulfed by eukaryotes between 2 billion and 600 million years ago, the cyanobacteria and their thylakoids have been fully incorporated into the eukaryote cells of plants and algae as fully dependent chloroplast organelles. The thylakoid membrane that separates the thylakoid lumen from the chloroplast stroma contains the whole photosynthesis electron chain, plus the ATP synthase motor that makes ATP from the proton H+'s gradient that will form across the thylakoid membrane once photosynthesis gets going. The photosynthesis electron chain is made up of photosystem II, PS2, a plastoquinone, a cytochrome, and photosystem I, PS1. The electron transport chain actions result in an increase in protons H+ in the thylakoid lumen. The proton H+'s gradient between the thylakoid lumen and the chloroplast stroma causes protons H+ to be drawn from the lumen through the ATP synthase into the stroma. The mechanical rotation caused by the protons H+ flowing through the ATP synthase rotor provides the energy to allow the ATP synthase to draw in ADP and phosphates Pi and compress them into ATP molecules. These ATP can be used to power the Calvin cycle that converts carbon dioxide and water into glucose. The photosynthesis electron transport chain is pictured in the figure. The photosynthesis electron chain includes two photosystem protein complexes and a cytochrome that transports the electrons between the two photosystems, PS2 and PS1. The two photosystems are named in order of their discovery. Photosystem II is the second photosystem discovered. It is named PS2, even though PS2 was later determined to be the first step in the electron chain. The name photosystem II stuck. Both photosystem II and photosystem I contain chlorophyll. In plants, the type of chlorophyll used in the photosystems is chlorophyll A. The figure shows the chlorophyll A carbon chain diagram alongside a ball and stick model of the chlorophyll A molecule. The chlorophyll molecule consists of a head and a tail. The long carbon chain tail is used as an antenna to catch photons. When any part of the chlorophyll is struck by a photon of the right wavelength, preferably a red light photon, the chlorophyll molecule gives up an electron to the electron chain. The loss of the electron causes that part of the chlorophyll molecule to become positively charged. An electron is passed up the chlorophyll molecule from the head to where the electron was lost. The head of the chlorophyll contains a carbon ring of oscillating single and double bonds. The carbon ring surrounds a magnesium ion that helps in electron transfer. The magnesium ion is held in place by 4 nitrogen atoms. Once the chlorophyll head gives up one electron, it becomes positively charged and desperately seeks a replacement electron. There are around 40 chlorophyll A molecules in the photosystem II protein complex. The chlorophyll A is affected by red light. Photosystem II has other molecules that are affected by other wavelengths of light. Beta-carotene will lose an electron to the electron transport chain if it is hit by a blue light photon. Green light photons are the least likely to be absorbed and are usually reflected off the photosystem complex. This reflection of green light is why leaves usually appear to be green. All the light-absorbing molecules in photosystem II, the chlorophyll and the beta-carotene, are connected to a pair of chlorophyll molecules at the thylakoid lumen side of the complex. This pair of chlorophylls is known as P680. 680 nanometers is the wavelength of red light that activates the P680 most strongly. The other light-absorbing molecules act as antennae for the P680 and increase the likelihood that an incoming photon that strikes anywhere on the photosystem complex will channel its effect to P680. On the lumen-facing surface of PS2 is another structure called the oxygen-evolving complex. The oxygen-evolving complex contains calcium Ca and manganese Mn bound in place by multiple amino acids. The oxygen-evolving complex holds onto a pair of water molecules as the P680 chlorophyll pair splits them into protons H+ and oxygen O2. A conceptualized model of the oxygen-evolving complex is shown in the figure. A reconstruction of the protein structure of photosystem II is shown. The chlorophyll A and beta-carotene antennas for the P680 chlorophyll pairs are shown in the figure. The P680 is not shown, but should be at the water oxidation site circled in yellow. The coenzyme plastoquinone Q is the electron acceptor for electrons lost by the antenna from photon hits. Plastoquinone is very similar to the ubiquinone, or CoQ10, that takes part in the electron transport chain of a mitochondrion. Once the plastoquinone Q accepts 2 electrons, the negative plastoquinone Q2 negative draws 2 protons H+ from the stroma and becomes plastoquinol QH2. The plastoquinol QH2 leaves the PS2 complex and transports its load of electrons and protons H+ to the electron transport chain. A recycled plastoquinone is ready to take its place in the photosystem II complex. Now that we know the structure of photosystem II, then its activities can be recounted step by step. In step 1, a photon of red or blue light hits the chlorophyll or carotene antenna. The photon knocks an electron off the chlorophyll or carotene antenna molecule. In step 2, the plastoquinone Q accepts the electron. When the plastoquinone accepts 2 electrons, it pulls 2 protons from the stroma and becomes plastoquinol QH2. The plastoquinol QH2 leaves the PS2 complex to join the electron transport chain. A recycled plastoquinone takes its place in PS2. In step 3, to replace its missing electron, the chlorophyll antenna pulls an electron from P680. In step 4, the P680+ needs a replacement electron, so it pulls an electron from a water molecule H2O held by the oxygen-evolving complex. In step 5, every electron lost by the water molecule causes it to release a proton H+ into the lumen fluid. In step 6, once the 2 water molecules held at the oxygen-evolving complex have lost 4 protons, all that is left is 2 bare oxygen atoms. The oxygen atoms bond immediately to become molecular oxygen, O2. The oxygen O2 leaves the chloroplast as waste. In step 7, the 2 water molecules are replaced in the oxygen-evolving complex. The 4 free protons H+ are free to migrate to the ATP synthase, where they can be used to make ATP. To recap the PS2 photosynthesis, every 4 photons cause the splitting of 2 waters and the release of 4 electrons to the electron transport chain and the release of 4 protons into the lumen. 2 H2O plus 4 photons results in 4 H plus and 4 electrons and 1 O2. The graphic shows the activities of photosystem II. To go on to the next step in the photosynthesis electron chain, we trace the path of the plastoquinol QH2. The plastoquinol QH2 carries its load of 2 protons H+ and 2 electrons from the PS2 complex to the cytochrome B6F protein complex. The B6F complex contains cytochrome B6, cytochrome F along with ferrous sulfide and other iron-centered heme groups, plus a plastocyanin waiting beneath the complex in the thylakoid lumen. The cytochrome B6F complex serves as an electron transfer body, much as cytochromes B and C did in the mitochondria electron transport chain. Upon contact with cytochrome B6F, the plastoquinol QH2 unloads its protons H+ and electrons into the cytochrome B6F. The 2 protons H+ are immediately discharged into the lumen. These 2 protons H+ came from the stroma, So in effect, the plastoquinol QH2 has acted as a proton pump to move protons H+ from the stroma to the lumen. The plastoquinol's 2 electrons take separate paths through the cytochrome. One electron attaches itself to the ferrous sulfide FeS, then moves through the cytochrome F, that in turn hands it off to the plastocyanin, a copper-containing protein. The plastocyanin will transport the electron through the lumen to photosystem 2. The second electron will be handed off to cytochrome B6, which transports it to the stroma face of the complex, where an uncharged plastoquinone Q molecule will accept it. A second plastoquinol QH2 arrives from the PS2 complex, and likewise unloads its 2 protons H+ into the lumen. One of its electrons goes to a plastocyanin to be carried to PS1. The second electron goes to the semi-charged plastoquinone SQ, and once the plastoquinone accepts the second electron, it uses its negative charges to pull 2 more protons from the stroma onto itself to become a plastoquinol QH2. The plastoquinol QH2 re-enters the thylakoid membrane to seek another cytochrome B6F to discharge its load of protons H+ and electrons. The figure shows the proton H+'s discharge into the lumen and the electron transport by the plastocyanin PC, but not the pickup of protons H+ by a third plastoquinone Q at the top of the cytochrome B6F complex. The next figure is a representation of B6F, or the cytochrome B6 and the cytochrome F protein complex. The next step in the photosynthesis electron transport chain follows the plastocyanin to the photosystem 1 protein complex. Photosystem 1 has many of the same functions as photosystem 2. It catches photons with chlorophyll and light-sensitive cofactors, but in a much larger complex. According to Nathan Nelson and Charles Yoakum, PS1 is composed of more than 110 cofactors, significantly more than photosystem 2. Photosystem 2 has 50+ cofactors, where photosystem 2 might have 30 to 40 chlorophyll antennas. Photosystem 1 often has 120 to 200 chlorophyll and 20 to 50 carotenoid antennas. Photosystem 2's focus is the chlorophyll pair P680. Photosystem 1's focus is chlorophyll pair P700. P700 is tuned to 700 nanometer light, making P700's optimum light source even redder than that of P680. Unlike photosystem 2, photosystem 1 has no oxygen-evolving complex that splits water. Instead, when the P700 loses an electron to its antenna, it uses the positive charge of P700+ to pull a replacement electron from the just-arrived plastocyanin. When an antenna is struck by a photon, it loses 1 electron to the iron-sulfur complex connected to ferredoxin Fd. The antenna molecule replaces its lost electron with one from P700. The P700+ pulls a replacement electron from plastocyanin to return to its neutral P700 state. In the stroma fluid, 1 ferredoxin Fd accepts 1 electron which it transfers to the enzyme ferredoxin NADP plus reductase FNR. A second ferredoxin Fd transfers its 1 electron to FNR. The enzyme FNR uses the coenzyme flavin adenine dinucleotide FAD to add the 2 electrons acquired from the 2 ferredoxins to convert a passing NADP+ and a passing proton H+ into a NADPH. The NADPH will be used along with ATP to power the Calvin cycle that the chloroplast uses to convert carbon dioxide CO2 and water H2O into glucose C6H12O6. NADP+ is nicotinamide adenine dinucleotide phosphate, a molecule identical to the NAD+ molecule that we saw in the citric acid cycle and mitochondria electron transport chain. Except NADP+ has 3 phosphates instead of the 2 in NAD+. In this next figure, NADP+ is shown alongside NAD+. Note: the only difference is the extra phosphate group PO4 in the NADP+ molecule. As we have seen, the photosynthesis electron transport chain has absorbed many protons H+ from the stroma fluid of the chloroplast and transferred many protons H+ into the lumen fluid of the thylakoid body. In addition, in photosystem 2, when the two water H2O molecules are split by 4 absorbed photons, 4 protons H+ are released into the thylakoid lumen. So, after one pass through the photosynthesis electron transport chain, there exists a large proton H+ imbalance across the thylakoid membrane between the lumen and the stroma. The only way these protons H+ in the lumen can get back through the stroma will be through the ATP synthase rotor. As we saw when we studied the mitochondria electron transport chain, the protons H+ flowing through the ATP synthase will cause its rotor to turn. And that turning rotor will cause the F1 motor on the ATP synthase to squeeze ADP plus phosphate P into ATP. The efficiency of ATP synthase is about 2.5 protons H+ per ATP, or about 10 protons H+ for every 4 ATP. These ATP, along with the NADPH, will be used to power the Calvin cycle, which is the glucose production stage of photosynthesis. The photosynthesis electron transport chain is often called the light-dependent reactions of photosynthesis. The Calvin cycle is often called the light-independent reactions, or dark reactions of photosynthesis. The Calvin cycle, or dark reactions, occur without any need of light photons. Despite the name dark reactions, the light-independent Calvin cycle occurs during daylight hours simultaneously with the light-dependent photosynthesis electron transport chain. The Calvin cycle takes place within the stroma fluid of the chloroplast. Before I get into the details of the Calvin cycle, I want to close the discussion of the photosynthesis electron transport chain and photosystems 1 and 2 with some discussion of the origins of photosynthesis. I will discuss the origins of photosynthesis in my next video.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.